And we're back. Welcome to Class 5. I think it's Class 5. If it's not Class 5, just forget what I said. So, I hope you all learned a lot from Esther and Jill while I've been gone. I was traveling around in China a little bit. Hey, what's up, everybody? How's it in North Carolina? Is it good? I lived in North Carolina for a little bit, actually. Boone, North Carolina. But today we're going to talk about plasmid DNA. And I think that's it. And do a demonstration. We're going to work with SnapGene. So I don't know if it told you to download SnapGene, but for the demonstration today and in general, you should have SnapGene. Let me pull up the URL. SnapGene Viewer, it's free. You don't need to use the SnapGene other thing. So there's the link. If you can't see the link, switch it from live top chat to live chat or something like that. Live chat to top chat. Just try them both until you see the link that I just posted for SnapGene. Get SnapGene. We're going to look at some plasmids, and we're going to talk about plasmids today and DNA and genetic engineering, because we're going to finally start getting into some genetic engineering experiments and working with genetically modified bacteria and organisms. Pretty cool. So after you learned, how to make plates, how to use a pipette, how to do all these different things, the next step is to use these tools to be able to engineer organisms. SnapGene Viewer did not work properly for you, Mr. Metric. Well, I'm sorry. There are other DNA viewers I'm sure you could find online. So if SnapGene Viewer doesn't work, you can look up and find other ones. It's just, I think it's the easiest to use one, and so I use it. You could just follow along with it, but you should be able to find other ways to look at DNA online. So it shouldn't be too much of an issue if you can't get it to work. But yeah, plasmids. Plasmids. So a plasmid is a circular piece of DNA, right? So plasmids are actually discovered in bacteria. They're naturally occurring. Bacteria have their normal genome, the big chromosome or chromosomes that their genome resides on. And then sometimes they have these little small pieces of DNA called plasmids. Now, plasmids are not part of the normal genome. They're not considered chromosomal. They're considered extrachromosomal, which is separate from the chromosomes. And they're usually pretty small. So for the most part, plasmids are a few thousand DNA base pairs. But they can be pretty big. They can be over 100,000 DNA base pairs. But generally, in genetic engineering, plasmids are kept in the thousands, maybe 12,000, 15,000 if you're really trying to push it, but you keep it small. That's because DNA is easier to work with, it's easier to manipulate, it's easier to put in organisms the smaller it is. So when you're genetically engineering your own plasmids for bacteria, you generally try to keep them pretty small. So you have a goal. You want to genetically engineer yourself. You want to genetically engineer some organism. Basically, the goal is to get DNA into cells, right? That is the goal. You just want to get DNA into cells. Because if you get the DNA into the cells, then the cells can theoretically use that DNA to do what you want. So the first goal of genetic engineering is to get DNA into cells. Now, generally in higher organisms, so animals and even plants, you use viruses or uh, organisms to get the DNA into cells. So viruses are great because they love to get inside of cells. So if you put in your own payload inside the virus, they'll take that and they'll put it inside the cell. But the way you can also get DNA in cells is you take a small piece of DNA, like a plasmid, and you just mix it with the cells. So you inject it into the organism, you maybe mix it with some chemicals that helps it get inside cells, but plasmids can go inside cells just on their own because they're small enough, and sometimes you can use some chemicals to help out. So sometimes you use things like calcium chloride, which neutralizes the charge of DNA, polyethylene glycol, which it's a polymer that kind of helps open up the cell walls. Use things like lipofectamine, which is like lipids, or use other polymers that basically help open up the cell walls a little bit to get the DNA inside. Basically, plasmids are usually small enough that you can convince some cells to uptake some of the plasmids. Now, in bacteria and yeast, plasmids are usually the main way to get DNA in cells. It's because bacteria and yeast, their cell walls aren't as robust as animals. And there's just a lot of bacteria and yeast, so they're easy to grow, easy to select, easy to subject to lots of chemicals and stuff like that. The cells are pretty robust, right? I mean, yeast and bacteria are everywhere. They grow in everything. So when you're genetically modifying bacteria and yeast, you rarely use viruses or anything like that. It's just not a thing that people tend to do. So you'll use plasmids. Plasmids, these small circular pieces of DNA, right? So we're going to take a look. If you got your SNAP gene, what we're going to do is we're going to take a look at what a plasmid looks like. What is a plasmid? What does it look like? What's going on with it? Let me pull it up on the screen. If I can. Where is it? Come on, Snap Gene. Show up for me. Capture cursor. All right. Let me try something else. Hold on one sec. Capture. Okay, there we go. Guys, that's way too big. Let me make it smaller. So you can see Snap Gene. All right. So what you're gonna do is you're gonna open up Snap Gene. So if you go to the Google Classroom, what you can do is you can download the plasmids that we have. So the plasmids on the Google Classroom should be bioinformatics material PD underscore GFP and bioinformatics material ProK1 EGFP. So we're going to look at the ProK1 EGFP sequence. So download that sequence and open it up in SnapGene. And you should see something like this. This is what we're going to look at. So get SnapGene, get it open, and load this up. Okay? So this is what they call a plasmid map. This is a representation and a map of all the different pieces of DNA that are in a plasmid. And they all do different things, right? So this plasmid, all plasmids, have three main parts. So the first part is what we call the origin of replication. Y'all following along with me? Everything good? Get that SnapGene up and running? So the origin of replication is what tells the cell that it should replicate this DNA, right? So you imagine if you put a piece of DNA in a cell, like a bacteria or a yeast, they replicate fairly often. So when they replicate, if the piece of DNA doesn't tell the cell to replicate it, it doesn't get replicated, which means every future generation won't have this piece of DNA. But what you want is you want every future generation to have this piece of DNA, so you need to put an origin of replication in it. This basically tricks the cell into thinking that this DNA is a chromosome or something that the cell wants, and so the cell replicates it for itself. Now, there are different types of origins of replication, but you don't really need to worry about that too much right now. Basically, these different origins of replication just help with the number of these pieces of DNA inside the cell. So you can imagine the more pieces of this DNA, the more effect your genetic engineering will have on the cell. So sometimes you want less, sometimes you want more, there's different amounts of things that you can deal with. So the second thing almost every plasmid has, is an antibiotic resistance gene or cassette. So this plasmid has an antibiotic resistance gene that confers resistance to ampicillin. You can see right there. Ampicillin, carbenicillin. Um, it basically degrades beta-lactam antibiotics. So it's a beta-lactamase. So what happens is the reason you want antibiotic resistance cassette. So when you're genetically engineering an organism, bacteria, yeast, whatever, only a percentage of the bacteria or yeast get genetically engineered, and it's usually a small percent. Maybe like 10% or something like that, right? So how do we know which bacteria or yeast or human cells get genetically modified? Well, there's a couple ways. One is you could try to search through all of them, and do DNA sequencing or some other crazy thing to try to figure out which ones have been genetically modified. Or what you can do is you can use a selection method to kill all the ones that haven't been genetically modified, right? So if we grow up the bacteria in the presence of ampicillin or carbenicillin, all the bacteria that don't have this plasmid are going to die. None of them are going to procreate and live. Only the bacteria that has this plasmid is going to live because it's producing this beta-lactamase gene. And this beta-lactamase gene is going to make sure that the antibiotic doesn't have an effect on that bacterial cell. So what we're doing is we're selecting against bacteria that aren't genetically modified by putting in these antibiotic resistance genes. Now, almost every plasmid has some sort of antibiotic resistance gene, right? So if it's a plasmid for yeast, if it's a plasmid for human cells, if it's a plasmid for dolphin cells, it has an antibiotic resistance gene. And this is because it just makes it so much easier to do genetic engineering. It makes it so much easier to see when the genetic engineering has actually worked, right? So almost every plasmid will have this origin of have this antibiotic resistance cassette in it. The F1 origin is some leftover. Um, so you have to understand that like a lot of plasmids have a lot of crap in it that they don't need. It's just because plasmids were created, a lot of them, the first plasmids were created a long time ago. And genetic engineering techniques weren't the greatest. So they just kind of slapped stuff together and hoped it worked. And it did, right? They slapped a bunch of stuff together. But as time went along, we started to see a little bit more about what was needed and what wasn't and stuff like that. But also scientists, they care less about what is the best plasmid and more about just does it work. So a lot of times you see stuff like this F1 origin, which is a bacteriophage origin. It doesn't really contribute to, the life of the plasmid. It's just some leftover thing that they used a long time ago. All right. So generally, the final thing that you see in a plasmid is the gene of interest. So when you're trying to engineer an organism, obviously you're trying to put some gene in it, right? You want to put in some new DNA that does something. So you take a plasmid. A plasmid is what they call a backbone. And this backbone contains the origin, it contains the antibiotic resistance, and then you put in your gene, and then you can use it in the bacteria you're trying to genetically modify. So you don't have to create all these things from scratch. You just basically have to get the gene and stick it in. Now, genes, these can be found in the organism. You can use techniques like PCR to amplify genes from organisms directly, or you can just have them synthesized. So there are a number of companies, including GeneWiz and other places, that you can go to, and you could ask them to synthesize the DNA for you. You just tell them what DNA bases you want, and they will synthesize it for you, and they'll send it to you. And after they synthesize it and send it to you, then you can stick it inside this plasmid using some molecular biology. So in this case, our gene of interest is the green fluorescent protein, right? So the green fluorescent protein is a protein that fluoresces green, oftentimes used to tag cells, to tag different proteins. Um, it's used in a large variety of things, and it came from a jellyfish. Yeah. So the green fluorescent protein, this protein is from a jellyfish. Now, what we can do is we can actually look at the sequence of this stuff. So when people found the green fluorescent protein, when they saw a jellyfish that fluoresced at night, what they were interested in is, well, how does this jellyfish do that? Let's see if we can isolate this protein, this gene that creates this protein from the jellyfish. So using some techniques In DNA sequencing, what they found out is they found the DNA sequence for GFP, green fluorescent protein, and they found out that you're able to stick it in basically any organism and it still works just fine, right? Now, this is the case sometimes. It's not the case all the time. But sometimes what you can do is you can take a gene that codes for a protein from one organism and you can stick it in another organism and it'll work just fine. This is just, so all life generally codes for proteins the same way, right? So it uses the same nucleotide codons to code for proteins. The same DNA to make the same protein. Doesn't matter what organism it's in. But now after it makes the protein, there are certain things that can be intrinsic to the organism. There can be things like, does the protein after it's made get modified by other things in the cell? Sometimes this happens. Sometimes proteins get modified after they're made and that helps them function, right? Sometimes it's just like the different components inside the cell. Sometimes these proteins interact with other proteins, they interact with other things, and if the cell isn't the same, right? So the inside of a human cell is obviously going to be different than the inside of a bacterial cell, or maybe not so obviously, I don't know. But they're going to be very different. Then sometimes a bacterial cell won't have the correct environment or the correct proteins to interact with this protein we're trying to put into it. Then what happens is it doesn't work. Nothing happens. It just doesn't work. But in a number of cases it does work, like in the case of green fluorescent protein, right? Green fluorescent protein works in a wide variety of organisms, including bacteria and yeast. So this is a plasmid for bacteria, and this is the green fluorescent protein DNA. That's what you're looking at. DNA for the green fluorescent protein. All right. How's everybody doing? So this plasmid has two genes. Alexis, it depends what you mean by genes. So this plasmid has one gene of interest. They call it a gene of interest when you put a gene inside, but it has other different genes, right? So it has like the antibiotic resistance gene. It has the origin of replication. Uh, so technically it could have more than one gene, more than two genes, depending on how you look at it. But generally, we don't refer to any of the genes on the backbone as genes. Those are just part of the the DNA backbone, the plasmid backbone. The only thing we refer to as genes are the genes we put inside. So if somebody were to say how many genes were on this plasmid, I'd say one. It's the green fluorescent protein. That's the only gene on this plasmid. Yeah. Mr. Metric, do we know what every single base pair is for, or are some parts of the plasmid a mystery? So we generally don't know what every single base pair is for. Some of it is kind of a mystery. I don't think there have been really any in-depth studies that I could find on plasmids. That's just because people don't care enough. It's not fancy. It's not exciting work. And stuff works. So why change it? Why try to figure it out? But there are a lot of different things in plasmids that have unknown functions or are there for unknown reasons. Um, things like spacers, which are just regions in between some of these DNA elements. Um, yeah, there's different stuff, different length origins, different stuff that are in these plasmids that might not necessarily have an effect we know of. One thing you can do is if you use, say, NCBI, National Center for Bioinformatics or something like that. NCBI, go there. NCBI Blast. So the program's called uh, Blast. What you can do is you can take a plasmid and you can compare it to everything else that's out there. So you can try to find out, like, are there some plasmids that exist without these pieces of DNA? Are there some plasmids that have this piece of DNA? You know, how does it compare? How does it contrast? You said because synthesis costs increase with length, shouldn't useless stuff be deleted? So plasmids aren't actually synthesized. So once you make a plasmid, generally what you do is you put it in a bacteria like E. coli. Once it's in E. coli, what you can do is you can replicate that bacteria by just giving it, the antibiotic needed for selection and just some food, right? Some sugar, water, LB broth or something. And what this does is it grows up a lot of bacteria and a lot of the plasmid DNA. From there, it's fairly easy to purify large quantities of plasmid DNA from the bacteria because you could just get a ton of the bacteria, right? Because bacteria are just so easy to grow. You did the experiment where you just grow bacteria from your hands and whatever. Like, bacteria is everywhere. It's really easy to grow. So once you make your original plasmid, after you synthesize the DNA for the original plasmid or synthesize the DNA for your gene that you put in your plasmid backbone, you don't have to pay anything else except for the cost of media to grow up the bacteria, basically, right? It becomes exponentially cheaper once that DNA is put in a plasmid and put in an organism. Yeah, it becomes pretty pretty inexpensive. So theoretically, if you make a new piece of DNA, you could distribute it to all your friends for inexpensive, reasonable cost. Shouldn't cost much at all. All right. So where were we? We were talking about genes, right? So now on these plasmids, there are many different DNA elements, right? So the DNA elements aren't just the gene and the antibiotic resistance gene and all this other stuff. What you're trying to do with the plasmid is you're trying to convince the cell to think this DNA is actually a chromosome or a part of its genome. So there are a lot of other things that you have to include to trick the cell into thinking that. Things like a ribosome binding site, right? So the ribosome helps turn the mRNA of this protein into the protein, right? Takes the mRNA, it builds amino acids together using the codons, and you get a protein from the ribosome. So if you're trying to get a protein, generally you're going to have a ribosome binding site. So you got that in there. On the other side, you also have things like terminators. So terminators basically tell the cell that it needs to stop making the protein or the DNA that you want, right? So a terminator is just another piece of DNA that helps trick the cell into doing things. So basically, you're just trying to hack the cell with all these different DNA components so that it thinks that your piece of DNA is actually a real part of its genome, right? And so you can see all through this how long the DNA is, where does it fit in the plasmid, and everything like that. You can see the sequence for all these things here. So a higher length, Mr. Metric asks, a higher length increases replication time. It doesn't actually. So when you're growing bacteria, you're actually usually growing them up over a period of like 12 to 18 hours. And so replication time has absolutely no effect on something like this, right? So let E. coli bacteria, its replication time at 37 degrees Celsius is somewhere approximately between 30 minutes and 60 minutes. And replication time is not really going to affect that too much, right? Not, and by that I mean that like you're not going to see a difference really. If you grow up two bacteria side by side and one has a slower replication time, you're not going to see much difference. You're still going to be able to grow up a lot in, one night for the most part. Obviously, not every situation, but for the most part, bacteria will just grow and increased replication time won't be too big of a deal. So if you insert gene of interest into plasmid and your gene of interest contains a terminate sequence in the middle or the beginning sequence, is there something to fix it? That's kind of tough. It really depends if there's restriction enzyme sites or not in the plasmid. So there are these things that scientists found are little enzyme proteins that cut DNA. These are called restriction enzymes or restriction endonucleases. Now in the plasmid, if you look at the plasmid map, what you can see is you can see all these with all these different letters and all these different numbers. Now, what these refer to are the places in the DNA where these restriction enzymes can cut the DNA, right? So the question is like, well, why would you want to cut the DNA? What does that do? Now, imagine if we are trying to remove this here from the DNA, right? We're trying to remove this from the DNA, and we need to figure out a way to do it. Well, the best way to do it is to just cut it out and remove it, right? So if we look on either side of this element, or approximately, we have SAP1 and BSMB1, right? So we got those two restriction enzyme sites. So if we look at the sequence right here, so we got SAP1. Now SAP1, it cuts DNA at this specific sequence right here, right? AAGCG. Or here's this, you can see right up here where, right, SAP1, something like that. Maybe a little too long. That is where it cuts DNA. And then we could go down here and we go to BSMB1 and we can see that it cuts DNA right down there. Now what we can do is if we mix the DNA with these enzymes, the enzymes will cut it, and then we could seal off that cut using other enzymes so that we can remove different elements from this circular plasmid piece of DNA. But still, it's even though it sounds like it would be kind of easy, it's still a little bit complicated. It's complicated in molecular biology because you can't see the things that are going on. So you just have to mix stuff together and pray for the best. At least that's what I do, right? Hope it fucking works. All right. So We went over restriction enzymes, went over all this stuff. Does anybody have any questions right now about plasmids, about genes, about restriction enzymes, about how plasmids are used? Hit me with it. Let me know. So, in the class, if you in the class for this week, you should have an experiment of taking this Pro-K1 EGFP plasmid and sticking it inside bacteria, right? So that should be part of your experiment for this week. So what you're going to do is you're going to take this plasmid DNA, you're going to mix it with bacteria and some chemicals. The chemicals will help the plasmid DNA get inside the bacteria. Then remember, you're going to take this bacteria and you're going to plate it on agar with antibiotic. The antibiotic will kill all the bacteria that haven't been genetically engineered, and hopefully the only thing you'll have left is bacteria with jellyfish gene in them that will fluoresce. Yeah, restriction enzymes are basically the foundation of modern molecular biology. As things start to move along and DNA synthesis gets cheaper, they become less and less important because companies will just synthesize a gene and put it in a plasmid for you for inexpensively. But basically, for the past, since the 1980s, maybe almost 40 years, they've been the foundation of molecular biology because they allow you to cut DNA and insert new things in, right? In a very specific manner. Yeah, so the direction arrows in the plasmid, if you're looking at the direction arrows, here, let's pull this back up. If you're looking at the different direction arrows in the plasmid, these refer to which strand is used to create the mRNA. So it's the directionality of the gene, right? So if we look at a sequence, so this gene, for instance, it is going this way, right? So what this means is that the strand that is actually being used to make it is opposite of what we normally think, right? So instead of the top strand, TAA, TTA, what's actually being used is the TAA below it, right? So it's just DNA can be used, either strand of DNA can be used to encode stuff, right? And one strand is not the same as the other, right? So TAA, TTA is not the same thing as TAA, right? They're different things. CCA is not the same thing as TGG, right? So DNA has a directionality to it. You can see up here, DNA has a directionality which they say goes from the 5 prime. The 5 prime refers to a specific molecule in the DNA base or backbone. So it goes from 5 prime to what they call 3 prime. And the bottom strand also goes to 5 prime to 3 prime. So the top strand up here, what you can see is it starts at the 5 prime and the bottom strand ends at the start. So it's the 3 prime. So this, the GFP for instance, right? It's going in the forward direction, so to speak. And this lets us know that it's using the top strand to encode the mRNA, the protein that gets made. So the top strand is what is being used to encode it. So it's the directionality of the DNA. Oh, bacteria uh, evolve antibiotic resistance all the time. Um, so what you can do, I mean, you could just take bacteria, wipe your arm, right? Take a swab of your arm and put it on a plate with antibiotics, and there are bacteria that will live. Yeah, like bacteria that are antibiotic resistant are everywhere. They're naturally occurring. They were around even before we made antibiotics, right? So it's the idea that antibiotics made bacteria antibiotic resistant is false, right? Though overuse of antibiotics can definitely lead to some bacteria evolving antibiotic resistance, bacteria are naturally resistant to some or many or all antibiotics. It's just naturally occurring, right? You can imagine if you have, you know, I don't even know the number of bacteria that have existed in the history of the earth, but it's been a lot, right? Way over a trillion. I don't even know. Google bacteria. Like obviously they're gonna evolve things that just make them antibiotic resistant or resistant to these antibiotics. What determines what strand is used in the DNA? That's just the DNA elements. Remember how we talked a little bit about other DNA elements like ribosome binding sites and terminators and things like that? Um, those things help determine what strand is used. Well, so you have to understand that DNA enzymes or things like that, they only move in one direction, right? So they only move generally from 5 prime to 3 prime. That's just generally the way it works. So they tend not to move in the reverse direction, though it's not always true. So when a polymerase is binding to the DNA and is determining which strand to use, it's only going to move in the one direction, right? It's only going to move from 5 prime to 3 prime on the top strand or the bottom strand. It's never going to move from 3 prime to 5 prime on either strand. Not never, but like take it as a general rule. All right. Any questions? Any questions about plasmids? Any questions about the experiment you're going to do? I'll just sit here and wait for the lag to catch up to make sure. Anything. Hit me up. Let me know what questions you got. Mr. Metric says, so it gets direction right often enough that errors don't matter. Yeah, it's generally going to get the direction right. It just, the majority of the time. Large majority. It can't read. The thing is that it can't really read the wrong way, Mr. Metric. It's not that it's going to read the wrong way. It just doesn't. Like the way that the structure of DNA and the way that the structure of these proteins and everything works, it doesn't work. So it doesn't read the wrong way generally. The large majority of the time it won't. And if it does read the wrong way, it'll just fall off. The protein will fall off, the polymerase will fall off. It won't continue to do it. So you won't really ever get a protein made, um, an mRNA made from reading the wrong direction, which will then become a protein. Yeah, think of it like a one directional sprocket, right? Or something like that. Like it can only go in one direction. If you try to take it in the reverse direction, it just doesn't work, right? It can only go in one direction. It's just because that's the way it was made. It's the same thing here. There's just only one direction it can go in. It just doesn't work. The structures don't line up. The physics doesn't happen. Okay, if that's the only questions, then I appreciate you all showing up today. As always, if you have questions about the experiments this week, genetically engineering the bacteria and other stuff, just let us know. Post on the Google Classroom. We're here to answer your questions. 5' and 3' is intuitive, Mr. Metric. It's intuitive to scientists because start and finish don't mean anything. 5' and 3' refer to specific molecules in the DNA, so it makes sense scientifically. Look it up. It'll make sense more if you understand the scientific premise of it. Um, it's going to be great. Exactly right. So get it done. Let us know. Show your results. Post pictures. Make sure you document everything and post pictures for us. And if you have any questions, post them on Google Classroom. Otherwise, we will see you next week. Hope everything is well.